Today we're going to look at Chapter 12 over plant structure and function. We're going to look at plant structure, the roots, the stems, the leaves, how plants transport water and nutrients, how they carry out photosynthesis, how do they do what they need to in order to grow, what signals, hormones, or chemicals are released, and how do plants respond to them. And we'll take a look at unique designs that plants have, some features that they utilize that allows them to live in multiple habitats. So, a brief introduction. What is anatomy and physiology? If that's what we're looking at for plants, we have to define it. Anatomy is the study of structure, where we're looking at what makes up an organism. What type of cell? How do they form tissues? Do they? We can also look at physiology, which is the study of processes, looking at the function of a tissue or cell or an entire organism. We can also categorize plant organs into two categories. Vegetative organs are not involved in reproduction, such as stems, roots, and leaves. The reproductive plant organs are involved in reproduction. They include things like flowers, fruits, and seeds. When we look at plant structure, we have to think about the small microscopic cells that make up these eukaryotic multicellular autotrophic organisms, because that's what plants are. They're made up of cells that have cell walls. These cell walls sometimes contain cellulose or even lignin. And these cells are specialized to perform certain functions, like those in leaves help carry out photosynthesis. We're going to see that the functions of plant tissue are going to be based on how they are grouped together, whether or not they are part of the root system or the shoot system, the two main systems of plants. The shoot system is composed of leaves. That's where photosynthesis takes place. And of course, we have to include the stems that support the leaves, as well as the stalk that supports the stems. Now, stems contain the tissue that transports substances between the roots and the leaves, whereas the root system supports the plant and absorbs water and minerals, and they are usually located underground. The root system consists of structures that anchor the plant and provide a place for food storage. So they both have their own structure, location, as well as function. We can also look at four basic kinds of tissue. Meristematic tissue is going to be cells that we might think of as stem cells. They are undifferentiated. These cells are not committed to becoming any specific type of plant cell. This would be our source of new cells that can become tissues, which can become organs. And so they can develop into any tissue the plant needs, and they would do so via mitosis. They are found in specific locations, like the tips of the roots and the tips of the shoots. There's also ground tissue, and there's three types of ground tissue. This is a very common type of tissue in a plant. It includes the parenchyma, collenchyma, and sclerenchyma. Now, parenchyma is going to be involved in photosynthesis. It helps carry out things like metabolism. Collenchyma is going to be made up of living cells, but they have uneven cell walls, oftentimes thickened by things like cellulose or lignin, and they're there to support young stems, roots, and petioles. For example, the strings you find in celery stalks are made of collenchyma tissue. Now, if we want cells that provide rigid support and protection, we'd look at the sclerenchyma. They have walls infused with a lot of lignin. They're found in the mature parts of the plant, and they're going to help make the ground tissue tough and strong. So they're definitely the most supportive. Structural would be collenchyma, and parenchyma is going to be more metabolic or photosynthetic. There's also dermal tissue, which is just an outer covering of the plant, like the epidermal layers of the leaves, both upper and lower, and this would provide protection, a shield, against the plant's internal environment and its surroundings. The dermal tissues of the root system are also responsible for absorbing water and minerals that the plant might need. So there are varying functions besides support and protect for dermal tissue. Now, what we're probably most interested in, and we'll talk a little bit more about, is the vascular tissue, which we didn't see in the bryophytes, like mosses or liverworts or hornworts. The vascular tissue is used in plants in order to carry water or nutrients throughout the plant, up the stem, past the petiole, to the leaves. The vascular tissue can transport water and nutrients through the plant, and think of these vascular tissues as essentially blood vessels. They're blood vessels for plants, and they're going to be synonymous with that of blood vessels in humans in function. There's two types of vascular tissue, xylem and phloem. Xylem is made up of non-living cells. Phloem is made up of living cells. Phloem carries sugar and organic substances, whereas xylem carries water and sometimes minerals dissolved in that water, and it will carry them from the roots past the plant's stem up to the leaves, and phloem also helps carry stuff throughout the plant. The dead xylem cells have a thicker cell wall than those that make up the phloem. Let's take a closer look at the roots. They perform three very important functions. They absorb water and nutrients from the plant surroundings and transport them to where they are needed. They anchor the plant. They're also used as a location for food storage. Most roots are below ground. They're in the soil, but there are two types. There are tap roots and fibrous root systems. Monocots tend to have fibrous root systems, and then gymnosperms specifically and many dicots tend to have taproot systems. So let's compare the fibrous root system to a taproot system. If you were asked to draw roots, what you would draw is probably going to look more like a fibrous root system than a taproot system. In a fibrous root system, the primary root begins branching very soon, and the system's going to look like an underground bush. Green onions would be an example of a fibrous root system. For a taproot system, we're going to have a primary root that is much thicker and will grow downwards, and it'll stay the main root, and you'll just have a couple of branches off that taproot. A carrot is actually an example of a plant with a taproot system. The carrot itself is the primary part of that taproot. We see that these root systems tend to have more surface area than even the leaves of the plant. And when a seed begins to sprout, it's actually the root that is going to form first, not the shoot. And the first root is called the primary root. Here we can look at a microscopic view of a root. We're looking at the root cap, which contains a meristematic region, an elongation region, and the maturation or differentiation region. This is a longitudinal cross-section of a primary root. We would just take a slice of the root along its length, and then we've looked at it under a microscope to gain this image. So you're looking at the side of the root. Now, those parts I just listed for the longitudinal cross-section of the primary root are described here. The maturation region is where the cells become fully differentiated. It's where they decide they're going to be root cells. They may have root hairs, and that helps increase their surface area. It allows even more water and nutrient absorption from the soil. There's an elongation region where we see cells begin to differentiate into specific kinds of cells. They begin to stretch out, and they fill their central vacuoles with water. The meristematic region contains undifferentiated cells that carry on mitosis. This area is, where we will reproduce or make more cells. This is where most of the growth of the root takes place. The tip of this region is actually called the apical meristem. It's the most active site of cell division in the in the root system. It enables the root to grow in length. And then the root cap is composed of dead, thick cell walls, and these cells protect the root as it shoves its way down into the soil. So here we see the zone of differentiation, zone of elongation, and the zone of cell division. And the zone of elongation is going to have the most mitosis. That's where most of the plant growth is going to occur in the root system. There is a zone of cell division. That's just where we have most of the meristematic tissue, and it's protected by the root cap. If you look at a lateral cross-section of a primary root, it looks a little bit different than that longitudinal section. So here we see a slice of a thin layer of the root, and that's from side to side or transverse. So we're looking kind of with a top view down the center. Now, Depending on if it's a monocot or a dicot, the arrangement of xylem and phloem throughout this root system will look a little different. You can see the dicot has this X in the middle, and then the monocot has a circle with another ring within it. The way I remember which is which is the dicot has an X, and that takes 2 lines, and the di in dicot means 2. And for the monocot, I see these circles, and I have multiple circles, but it's still only one shape. So that is just a circle. Whereas if I apply that same information up here, I have a circle and an X, that's 2 shapes. So different ways to remember dicot: an X is 2 lines, there's a circle and an X, that's 2 shapes. And down here I just have circles, so that's 1 shape, so that's a monocot. We can look more at the root and talk about things like the epidermis or the cortex or the endodermis. But these are all just different types of tissue you find within the root. If you look, for example, at the cells that are near the center of the root, you'd find that they tend to be near or around the xylem and phloem. That's because that endodermis, or that cell tissue near the inside of the root, is going to be made up of structures that are protective for these vascular cylinders that are going to be important for moving water and nutrients throughout the cell. Near the endodermis, you find things like the pericycle, cells that are undifferentiated and can form new branches of the root if needed. They would just break out from the endodermis to do so. And the root itself was protected by that epidermal cap. We see that around even the newest branches of roots. And the very central part of roots is considered the cortex, and that would be where substances are going to be stored for later use. Again, in the root of a dicot, the xylem forms an X shape. In the root of a monocot, the xylem forms a circle, or many ovals or kind of rounded structures. So that's a big difference between monocot and dicot, xylem and phloem. And we're specifically looking at the roots. We find in the monocot there's a pith, or spongy cells in the center. And inside them, the pericycle would be located. We never see a pith or these spongy cells in the dicot. All right, so here is the base structure for a monocot stem lateral cross section. So this is a stem lateral cross section, but you can still see this space where you would have kind of very spongy light cells. And this pith is only seen in monocots, never in dicots. Here we have a dicot stem lateral section. You can see we also have the X shape, and we have no pith. So if we're looking at the monocot and dicot vascular bundles up in the shoot, we can see that the arrangement is still going to contain the phloem and xylem, or the vascular bundles. But their arrangement differs. For the monocot, you're going to have scattered xylem and phloem. For the dicot, it's going to be arranged in a circle. In this case, in the actual stem, we're going to see a pith in the dicot and none in the monocot. If we take a close look at the phloem and xylem in the monocot and dicot, they look different. For the monocot, it kind of looks like a monkey face. For the dicot, it looks kind of like a mushroom. And when we look at stems, we also have to acknowledge that there are different types of stems. They have a variety of forms. They can be tall, they can be of trees and flowers. They can be kind of to the side, or like a vine, or prostrate, like in watermelons and cucumbers. They could be underground, such as we see in the potato. They can also be woody or herbaceous. Compare, for example, the stem of a flower to that of a tree. But regardless of the variety, they serve 3 basic functions in a plant. They support and manufacture the plant's leaves, conduct water and nutrients to and from leaves, and they carry out photosynthesis. Specifically in the leaves, but in some plant stems would only carry out photosynthesis when young, and then in others, they would carry out the photosynthesis for the entire life of the plant. So looking at stems, we have to acknowledge structures like the apical meristem. That's going to be where mitosis occurs for the shoot system or stems. And that's the top region of the stem. And this is an area of cell division or even more growth in the plant, like we saw with the root system. And the apical meristem is located in a structure called the terminal bud. So the terminal bud is where the apical meristem is located. The apical meristem is great for mitosis or further growth of the shoot system. The lateral buds that develop into new branches would grow out of the stem and produce new leaves. And when a plant grows in length, whether it's from its roots or its shoots, it is called a primary growth. There would also be nodal buds that could perform mitosis and produce new petioles that would have leaves. But here we can look at the difference between herbaceous versus woody stems. You can see a herbaceous stem on the left, a woody stem on the right. And looking at their vascular bundles is going to give you the biggest clue. We have ring-shaped xylem and phloem in the shoot system for woody stems, and we have scattered monkey face looking xylem and phloem in the herbaceous stem. Looking at herbaceous stems, we do see some similarities in monocots and dicots. Regardless, they do both have an epidermis. They both have vascular tissue, meaning they have xylem and phloem. They have a center area or cortex. The location of that cortex does differ, and that cortex tends to contain photosynthetic cells and store starch. In monocot stems, the vascular bundles were located in a scattered arrangement throughout the stem, whereas in the dicot stems, they formed a ring with a central pith. Another difference between both the monocots and the dicots is whether or not the cortex is found between the epidermis and the vascular bundles, or is it found throughout the stem. In monocots, it's found throughout the stem. In dicots, it's found between the epidermis and the vascular bundles. So here we see that the epidermis will be on the outside. For the monocot, we have scattered arrangement. For the dicot, we have a ring arrangement for all the vascular bundles. And again, in a dicot stem, the cortex is found between the epidermis and the vascular bundles. So the cortex would be right here or around the center, where we can't see a single area where we specifically find it thanks to the scattered arrangement in the monocot. So here's a quick review. We have an herbaceous dicot stem on the left and a monocot stem cross-section on the right. For a dicot, we have the mushroom shape for the xylem and phloem in the stem, and it's in a ring. For the monocot stem, we have the monkey face xylem and phloem arranged in a scattered composition. So let's not forget how the vascular bundles look. Again, in monocots, they look kind of like a monkey's face, with the phloem forming the forehead of the face. Think monocot monkey face for stems. And for the dicot, there is no illusion of a monkey face. We see the xylem clumped on one region and the phloem clumped in another region. It kind of looks like a mushroom. And mushrooms would have two parts. They would have a stalk and a cap. That's one way to remember it, the dicot. Woody stems do differ from herbaceous stems. We looked at the base structure a couple slides back. But if you focus on woody stems, we can look at the two layers that they are composed of, both an inner and outer bark. The inner bark is composed of vascular tissue and cortex tissue, or phloem and cortex tissue more specifically. There's also the cork cambium, which is between the inner and outer bark, and this layer continually produces cork cells that die quickly, and they would be impenetrable to water, gases, and most parasites, and they would form the more protective outer bark that we're more familiar with. Monocots would not produce woody stems, only dicots do. So we could look at dicots that have woody stems but never monocots. In a woody stem, the phloem is always near the outside of the stem. The xylem would be more inner or internal, and the phloem would become the inner bark. The xylem would become the wood. Because there's really no limit to the growth of a woody stem, new xylem and phloem would have to always be produced inside the stem, and that would be done by tissue called the vascular cambium. So it produces the new xylem and phloem as the woody stem or plant grows. It would produce the phloem on its outer side and xylem on the inner side. And this back and forth production of vascular tissue is what produces the rings in a tree, called the annual growth rings. You can actually tell how old a tree is if it's cut, and that would be by counting the rings. But you could also see some other information about general weather conditions during each year it was alive. There's also a growth in width, not just length or how tall they become, that we would call secondary growth, and that would only occur in woody plants, that thickening. Here we can see some labeled annual growth rings. That would be in a woody stem of a tree. And then let's also look at some of the specialized stems, like the ones underground, like in an onion plant. They would have bulbs, and the bulbs are not roots. They are actually a collection of underground leaves that sprouted from an underground stem, so they are a part of the shoot systems. Because they're underground, they can't perform photosynthesis. There's no light underground, so they're instead used for storage. Tubers, like potatoes, would be an example of an underground root or shoot system that looks like a root. And tubers are underground stems, essentially, that store excess food. The food they store tends to be in the form of starch. And that is it for the first look at chapter 12. And so we're going to stop there. Make sure to read up on the different types of plants. Go back and review what does xylem and phloem do, and what do they look like in the root systems versus the shoot systems for monocots and dicots.